All right, so today we are jumping into a really powerful scientific technique called the Western blot. But you know what? Forget the complicated name for a minute. Just think of this as a detective story, a cellular whodunit, where our job is to find one specific culprit. So here's the central question. Picture a single human cell. Inside, it is just a chaotic soup of about 20,000 different kinds of proteins all bumping into each other. Our mission is to find just one specific protein in that entire mess. I mean, how in the world do you even start to do that? Well, that's the case we're going to crack today. Okay, first up, a cellular whodunit. Before we can even dream of finding our target, any good detective knows you have to understand the crime scene. So let's set the scene for our investigation and see exactly what we're dealing with. And the scale of our challenge is just immense. We are talking about 20,000 different proteins all mixed up together. Each one's got a different job, a different size, a different shape, and our target, our culprit, is hiding in there somewhere. Just one face in a massive, massive crowd. So this is our secret weapon, the Western blot. You can think of it as our complete forensic kit, and it's designed to do one thing really, really well. Cut through all that noise to answer 3 simple but absolutely critical questions. Number 1, is our suspect even at the scene of the crime? Number 2, how big is it? And number 3, how much of it is there? Are we looking for a lone wolf or a whole gang? All right, on to our next section, the protein lineup. So what's the first thing you do in any police procedural? You round up all the suspects and put them in a lineup. We're going to do exactly that, but instead of lining them up by height, we're going to line our proteins up by size. And here's how we pull that off. Step 1 is lysis. We basically just blast the cells open to release all 20,000 of our protein suspects. Then comes denaturation. This part is super clever. We use a chemical that makes every single protein unfold and gives them all a uniform negative charge. Now, the only major difference between them is their size. We've leveled the playing field. Finally, electrophoresis. We apply an electric current, and all those negatively charged proteins start moving through a gel that acts like a sieve. The little, lightweight proteins zip right through, but the big, bulky ones get tangled up and move really slowly. And just like that, we have a perfect lineup sorted from smallest to largest. Okay, so we've got our lineup. But there's a problem. It's sitting in this flimsy, jiggly gel, which is not exactly ideal for an investigation. We need a permanent record. Think of it like taking a high-res mugshot of the entire lineup so we have something solid to work with. So here's the deal. The gel is fragile, and it's really hard for our witnesses, and we'll meet them in a second, to get in there and do their job. The solution? We transfer the entire lineup onto a sturdy membrane. It's kind of like a special piece of paper. We use electricity to blot a perfect mirror image of our proteins from the gel onto the solid surface. Now our evidence is stable and totally ready for interrogation. So we've got thousands of suspects laid out on our membrane, all in order of size. Now it's time for the most critical part of the whole investigation. We bring in our star witness, the one who can pick our single culprit out of that massive crowd. And in biology, that star witness is the antibody. And this interrogation is a careful 4-step process. First, blocking. We wash the membrane with a protein solution. And believe it or not, this is often just plain old milk to cover all the sticky spots so our witness doesn't get distracted. Next, we introduce the primary antibody. This is our highly trained specialist. It's engineered to recognize and bind only to our target protein. But we can't see it, so we send in backup, the secondary antibody. This one is carrying a tiny signal flare, like an enzyme, and its only job is to find and stick to our primary antibody. And finally, visualization. We add a chemical that acts like a trigger for that flare, causing it to light up or change color. And suddenly, our hidden suspect is revealed. And that brings us to the grand finale. Section 5, cracking the case. After all that prep work, the lights in the lab are low, we're developing our film, so to speak. We're about to see if our witness found its target and if our investigation has paid off. And there it is, the aha moment. A single glowing band appears on the membrane. That band, that is our culprit. It's the proof. It confirms our protein is there, and its position in the lineup tells us its exact size. Case solved. We got him.
So we found one protein in a soup of thousands. Big deal, right? Well, this is why it matters so much in the real world. That one glowing band is not just some lab result. It could be a biomarker telling a doctor a patient has early-stage cancer. It could be the proof a new experimental drug is actually working. It's the gold standard confirmatory test for diseases like HIV and Lyme disease. Every single one of these bands tells a critical story about our health. Now, the Western blot isn't the only tool detectives have. There's another technique called an ELISA. Think of an ELISA as a really fast way to do a head count. It can tell you if your suspect is in the room. But the Western blot does something crucial that ELISA can't. It also tells you the suspect's size. It's like not only knowing someone's in the room, but also confirming they're 6'2", just as described. That extra piece of data, the size, adds a whole other layer of certainty. And really, this quote just says it all perfectly. A Western blot doesn't give you a hint or a maybe or a clue. That single glowing band is definitive. It is the smoking gun evidence that proves beyond a reasonable doubt that your protein is there and tells you exactly what it looks like.
And so our investigation comes to a close. We've seen how this amazing technique finds one culprit in a crowd of thousands. And it really leaves you with a final, pretty exciting question to think about. If one simple glowing band can diagnose a disease or prove a cure works, what other huge medical mysteries are out there just waiting for the next Western blot to come along and solve them?